I am Wei. I'm a faculty member at the Computational Biology Department at Carnegie Mellon University, and my research focuses on using machine learning and computational methods to understand complex human diseases. And in particular, I am interested in understanding asthma and also how people react to drug abuse. Actually, for asthma, it's very heterogeneous disease. So that's also why we want to study this, because if you look at asthma patients of similar severity levels, they can respond very differently to the same treatment. And also, if you look at their asthma symptoms, their immune response profiles, they look very different. So all this heterogeneity makes treating these patients very difficult. So currently we're using this unsupervised learning approaches to understand subphenotypes of asthma, so that hopefully we can make treatment of this patients easier in the future.
Mm-hmm.
So the data we look at is really clinical data and also gene expression microarray data. As I mentioned, we use the machine learning approaches to look at the patterns of, for example, different drug responses in the asthma patients. And so a nice thing about machine learning approaches is that they actually allow us to detect patterns in asthma patients.
Mm-hmm.
And also, same thing for people abuse drugs. We also use similar approaches to look at how different people actually react to different drugs of abuse. Yeah, this is a good question. So, in the old days, clinicians can really just look at a few variables. They look at the lung function variables, they look at asthma symptoms, and they also look at what medication these patients actually take. So, these are all they could look at. But with the new machine learning approach, a nice thing is that we can now deal with a large amount of data, and also not only large in terms of the number of the patients, but also large number of measurements, and taken from all these different variables, which include also, for example, environmental variables, genetic variables, and also like inflammatory and immune profile variables.
Mm.
The reason is because we need to deal with different types of variables. For example, when I talk about this a large amount of variables, we need to deal with continuous variable, we need to deal with categorical variables, and also nominal variables. So the question is always how to put all this together computationally. And another thing which is very complex is the variables, which actually should be included in this kind of analysis. For example, there are always the irrelevant noise, or we call them noise variables. So it's always the question which variables to include, which can help us to identify clinically meaningful patterns.
Mm.